So in the last class, what we have talked about is we have started a new chapter that is called as strategies for enhancement of for food production, right? And so what we have talked about at that time is, just for a revision sake, what we remember is because of the ever increasing population, right, there has arisen a very serious problem, and that problem is of shortage of food. And to solve that problem, science has come up with various techniques or methods, and among the methods are animal husbandry, plant breeding, right, tissue culture, etc. So what we said is animal husbandry would be dealt in your zoology class. We have started with the point called as, or the topic called as, plant breeding, right? So what was plant breeding? We breed two plants, or we cross two plants, or we select two plants, and then if we want to have a plant with the desired traits, no, whatever we want, or we want to have a plant with improved qualities, what do we do is we go for such type of programs, right? Next, plant breeding korute, what we remember is there are certain steps of plant breeding, and what were the steps we are discussing? We have discussed the first step and the second step. What was the first step? In order to do, or in order to go for plant breeding, the first step was collection of what? Variability, right? And within that point we have discussed a word called as germplasm, right? So in order to study something, or in order to develop a new plant, the first work that you have to do is you have to collect all the alleles of all the genes that are present in the crop. And while taking or while collecting the germplasm, the point that we remember is it could be from within the country, or it could be from outside the country also. We had some words called as indigenous collection and exotic collections, right? After collection, what next? The second point was, now it's the time to evaluate and select the parents, right? So how will we select the parents? We will be taking two parents with desired traits, we'll cross them, and then we'll produce a new progeny. We were discussing point number 2, right? While we are talking about selecting, we have talked about the different methods of crop improvement. Crop improvement or different methods ki asile. There were 4 methods. The first was selection. Yeah, the second was hybridization, mutation breeding, and polyploid breeding, right? Yeah, these 4 were the 4 common points to which we will be talking about the methods of crop improvement. Selection we have completed. 3 types of selection we have said about, right? Mass selection, pure line selection, and clonal selection, right? And these 3, within these 3 headings, what we need to remember is je kuntu selection, kuntu type of plant, self pollinated ne, cross pollinated ne, vegetative reproductive plant, kunbilak plants or kuntu type of selection kora jay. For example, if you are talking about cross pollination, then the most common method of selection is obviously mass selection. If you are talking about, yeah, if you are talking about vegetatively reproducing plants, then obviously it's clonal selection. And similarly, self-pollinating plants, it's something that we have used that word is pure line, right? Pure line or inbreed selection. So these are the points that we remember from that point. The second is what we have discussed is hybridization. What is hybridization? The process of forming new hybrids, right? By crossing 2 different parents is called as hybridization, right? We have talked about that first natural hybridization, and the first artificial hybridization was seen in, I think you all remember something called as Fairchild, right? Fairchild mule bully eta asile, that is something that is considered as the first artificial hybridization, right? And the last point that we have talked about hybridization is hybridization, no doubt it sounds so good, na? It sounds so easy that you just select 2 plants and then you cross within them and then you produce the progeny, and then ultimately you get a new plant of your desired traits. But exactly, it's not so very easy, right? Enekwa nohoy je cross kora loge loge will get a plant that has the entire qualities. It goes for experiments and repeat and repeat. Tamane ki hobo, he hybrid to aru juntu progeny ami produce korilu, he progeny needs to be grown for various growing seasons, and then only after many years we will be obtaining that pure plant with our desired characteristics. So obviously it calls for some time. Right. So today we are continuing about that improvement methods only. So the third point through which, the third point that we are going to discuss today within the heading improvement of or crop improvement is mutation breeding, right? Mutation breeding. So here what we need to remember is we have to talk about what is mutation. We are not going to talk about it in details because this is something that will be dealt in your genetics class, but just to have a rough idea, what is mutation? Mutation boli kole ami ki buzo? What is mutation? It is a sudden, right, sudden change, stable change, and inheritable. Right? Inheritable change, and because that leads to what? That leads to change in gene structure, change in gene or change in gene structures, right? So this is what is called as mutation. While we are talking about mutation, we need to remember some other terms. The first is induced mutation, right? So mutation porhile, induced mutation is something that you have to remember. What is induced mutation? Induced means what? You do something yourself, that is what is called as induced, or you do something artificially, that is called as induced. Man-made mutation ok ami ki boli koi induced mutation. Naturally hoise ne manhuye kori dise. So manhuye kora mutation tok ami ki boli koi induced mutation. So the mutation that is done artificially. And how will you do it? Artificially, by certain physical and chemical agents is called as induced mutation. So ami artificially mutation to korisu, and or we are changing the gene structure of that plant by applying certain physical and chemical agents, and this is what is called as induced mutation. The third point is we have to talk about mutation breeding. Now what is mutation breeding? Induced mutation use kori palai jedi ami breeding koru, that is what is called as mutation breeding. So what is mutation breeding? Application of induced mutation for crop improvement is called as mutation breeding, right? So we had an idea the mutation ki hoy, induced mutation ki or mutation breeding, right? Since we are talking about mutation breeding, now we know this is something that is done artificially, and while doing artificial things we'll obviously need some agents of mutation, right? Kisuman physical agent lagibo, kisuman chemical agent lagibo, juntu niki ami apply kori pelai plant dalot mutation koribo paru, right? So we will be discussing a term called as mutagens. Have you heard the word called as mutagens? Mutagens ki bostu that induces mutation, right? So the things that bring about mutation or the agents that bring about mutation is called as mutagens. Agents that brings about mutation, right? And this agent, as I have said before, it could be either physical or it could be chemical. So it could be physical or it could be chemical, right? So if you are talking about physical, what would be the agents? For example, X-ray, right? UV ray. So if our organism is exposed to X-rays and UV rays, as a result what might happen is mutation might take place, and that is the reason why we are often advised by the doctors that we should not expose our body to X-rays more frequently, right? So he karone amak ki koy je X-ray besike koribo nalage, X-ray korile ki hoy je amar ki hobo je mutation hoar eta chance thake, because that might act as a physical agent or it might act as a physical mutagen. Similarly, if you are talking about chemical, nitrous acid, then acridine dyes, etc. could also act as a mutagen. So ebilak bostu jodi ami plant or ba cellot apply kori diu tetia hale ki hobo taharo gene or structure to they can get changed, and that change is all obviously called as mutations, right? So nowadays what we do, we use the help of mutation for the improvement of crop, because this is something that is really scientific and it requires less time also as compared to the longer procedure, right? So আমি select কৰিম, cross কৰিম, আকৌ তাৰপিছত grow কৰি পেলাই চাম, then we'll test whether the progeny has the desired characters or not. Instead of going such a long process, what we can do is we can directly go for mutation breeding. We need to remember certain examples of crops that has been developed due to mutation breeding. And if you remember, the day when we have started this chapter, what I said is, within this chapter what is most important is, sobotse besi important portion ki etu chapter, that you have to remember all the examples, no? Rice variety examples monot rakhibo lagibo, wheat variety examples monot rakhibo lagibo, right? So we have to remember the examples very properly. So examples of crops developed during mutation, there can be so many various types of crops, but we will be discussing only few of them. We would be talking about like Sharbati Sonara. Next, Pusa Larma. Then we could talk about Remy, Atomita, right? And then we can talk about a variety called as Aruna variety. So if you are talking about Sharbati Sonara and Pusa Larma, what we are talking about is something about wheat varieties, right? So questions kene kya hai? The question to ahibo pare, Sharbati Sonara or Pusa Larma is a variety of wheat, rice, castor, millets ne kihor. So you need to be proper that these two are nothing but wheat varieties and developed because of mutation breeding. Another point about them that you need to remember is they have formed the basis of green revolution. Formed the basis of green revolution. Green revolution is something that we will discuss, right? So Sharbati Sonara, Pusa Larma, ebilak kihere develop korise mutation breeding. Similarly, if you are talking about certain rice varieties also, these are examples of rice varieties developed because of mutation breeding. Aruna variety of castor. So these are few examples of crops that are developed according to or due to mutation breeding. So this is the amount that you have to know about mutation breeding within this chapter. As far as the process of mutation is concerned, details is something we will discuss during our genetics class, right? So this was a third method of crop improvement. We have talked about selection, we have talked about hybridization, the next is mutation breeding, and so finally the fourth is something called as polyploid breeding. Right? So this is the fourth method. We have a term called as polyploid. Now what is a polyploid? Polyploid ki bosto? We have often heard certain terms called as haploid, diploid, right? So what is haploid, what is diploid? To get a very simple answer, haploid means what? The organisms that has one set of chromosomes, that is called as haploid. The organisms that have two sets of chromosomes is called as diploid. So any cell that has two sets of chromosomes, one from the father, one from the mother. So that type of arrangement of two sets of chromosome is called as a diploid. Similarly, now we have a term called as polyploid. What is polyploid? Haploid buzilu, diploid buzilu, polyploid ki bostu? Any organism that has more than two sets of chromosomes, that is called as a polyploid, right? Organisms having more than two sets of chromosomes are called polyploids. Next is we have a term called as induced polyploidy. Induced polyploidy is used for crop improvement. Right? So, polyploid ki bostu we know. Now we are talking about induced polyploidy. Induce is a word that we have discussed while we were talking about induced mutation also. Induced mane ki something that is done artificially or man-made, that is what is called as induced. So, induced polyploids means what? We are making that organism, or we might be transforming our organism from a diploid to polyploid. So, আমি নিজে কৰিছো দুটা set of chromosomes আছিলে আৰু তাৰ পৰা আমি কি কৰিছো increase কৰি পেলাই more than 2 sets of chromosomes এটা cellot we have made that, and that is what is called as induced polyploid. And why we are doing this induced polyploidy আমি কিহৰ কাৰণে কৰিছো, but the main aim is for crop improvement, right? There are certain crops in which whenever the chromosome or particular sets of chromosome is increased, their yield or their quality or their taste or their height increases, right? Quality increase koribo pare, quantity increase koribo pare. So, eblak jodi ami artificially increase koribo bisariso, so we can either go for any of the 4 methods that we have discussed. We go for mutation breeding also, we can go for polyploid breeding also, right? Next, polyploid could be of 2 types: autopolyploid or allopolyploid. Again, details obviously in your genetics class, but to have a rough idea, what is autopolyploid? Increase in the same genome is called as, if I give an example, it will become clear. For example, we would be talking about a term called as triploid. So, what is triploid? Anything that has a structure, right? What we have seen is there is a gene called as A, and this gene has been repeated 3 times, and so it is called as a triploid. Similarly, if we talk about tetraploid, so we have one gene that we have increased it how many times? 4 times, right? So, what we have seen is it increases in the same set of genome. Eketa bostu A jedi increase korise, A increase korise, right? So, we are not talking about A and B duta gene or kotha koa nai. Eta gene jedi we keep on increasing and increasing. That is what is called as autopolyploid, and this technique has been used in plants like rice, wheat, maize, etc. right? So, enukwa dhoroner plants to increase their chromosome number, what we have used is we have used the technique of autopolyploid. Next, we come to the process called as allopolyploid. In this, what happens is polyploids are formed due to hybridization between 2 parents you can write, or 2 species you can write, followed by doubling of chromosomes, right? So, in case of allopolyploid, what we have done is duta parents select korisu, and what duta you can write either parents, and if this parent is confusing you, then you can write 2 species also, right? We are taking 2 different species, and what we have done is we have tried to make a cross between the 2 species, and the progeny that we will get after that, what we have done is we have doubled the chromosome of that F1 generation also. We will be writing down the steps. As of now, whenever we are talking about polyploidy or we are talking about polyploids, we know the number of chromosomes has been increased. Normal jiman asile tatke besi increase hoy gol, that is what is called as polyploid, right? 2 sets of chromosomes thakibo lagisile, etia besi ase, that is what is called as polyploid, right? Now the question is, how will you make it, right? So in polyploid বা doubling of chromosomes, chromosome number to যেতিয়া double কৰিবা তেতিয়াহে polyploid will form. এটা কথাটো হ'ল যে আমি double কেনেকৈ কৰিম? Do you remember anything from the chapter called as cell, where you have studied the different phases of cell cycle বা steps of mitosis, meiosis we have studied, right? So at that phase আমি যেতিয়া mitosis and meiosis we have talked about, do you remember the different stages of mitosis? Mitosis of first stage কি আছিলে? Prophase. After prophase that was metaphase, then anaphase, and then we had telophase, right? So what were the things that occurred in prophase? No, prophase, metaphase, telophase, anaphase. Prophase, metaphase, anaphase, and telophase. What we remember, there is one specific characteristic of each phase, right? For example, if you talk about prophase, what we remember, that thinning of the chromosomes, right? Disappearance of the nuclear membrane and the nucleolus. Enukwa kisuman amar points monot thake. Thik teneke, if you talk about metaphase, what is that one line that you remember? Unique characteristic of metaphase ki hoy? Either we are talking about mitosis or we are talking about meiosis, because metaphase is something that is seen in both the cases. So metaphase is a unique point to keep. What happens in metaphase? কি হয় মেটাফেজ? প্ৰফেজত যেনেকৈ আমি ক'লোঁ নিউক্লিয়াৰ মেমব্ৰেইনডাল নাইকিয়া হৈ যায়, নিউক্লিয়লাছ নাইকিয়া হৈ যায়, ক্ৰমোজোমছবিলাক কি হৈ যায়? যে ইট থিকেন্স, ৰাইট? মেটাফেজ, the chromosomes gets arranged in the equatorial plane. বা কি হয় গোটেই যিমানবোৰ চোৱা এটা সেল আছিলে, সেলটোৰ ভিতৰত নিউক্লিয়াছ আছিলে, নিউক্লিয়াছৰ পিছত ইয়াতে কি আছিলে? ক্ৰমোজোমছ বিলাক আছিলে. যেতিয়া নিউক্লিয়াৰ মেমব্ৰেইনখন নাইকিয়া হৈ যাব, then what will happen? The chromosomes will start segregating, or it will start what? Getting dispersed, no? Kute cell to mazote arrange ho jabo. So in case of metaphase ki hoisile, we had this, and all the sets of chromosomes ki hoisile the equatorial plane properly arrange ho gosile, right? So this was the characteristic of metaphase. After metaphase we have something called as anaphase. Anaphase ki hoisile, what is that line that we need to remember? In case of anaphase, splitting of the centromere. Do you remember something called as centromere? Right? So splitting of the centromere, simply what we can remember is anaphase was a stage when the chromosomes got separated to the two different poles, right? Pothomote gutei cell to asile tapsor mazot arrange hoy gosile, that is called as metaphase. After that this part went here and this part came to this pole, this is called as anaphase. Ar telophase ot ki hoisile, যে once the chromosome reached the two different poles, they got their nuclear membrane back, nucleolus akko arise hoy gol, and that is what is called as telophase. Now the question from this part is, when we are talking about doubling of chromosomes, what we need to remember is, if we apply a certain type of chemical at a certain stage of cell division, then we can increase the number of chromosomes of that cell. Generally কি হয় যে ইয়াতে যিমান ক্রোমোজোমস আছিল আলটিমেটলি দুইটা সেপারেট কৰিব আৰু দুটা নিউক্লিয়াই ফর্ম কৰিব, রাইট? কিন্তু তুমি যদি ক্রোমোজোম নাম্বাৰটো ডাবল কৰিব বিচাৰিছা, কি এপ্লাই কৰিব লাগিব? I think you all have heard something called as colchicine, right? So colchicine is a chemical which is generally used for increasing or doubling the number of chromosomes. Now the point is that ami kuntu stage of cell division or colchicine delay na chromosome number to double hobo. These are the extra points you have to know, right? So first point that we have understood today is, if you really want to double the number of chromosomes, you can apply colchicine from outside, right? Colchicine code dim, obviously cell or dim, but hey nucleus to dim. Kintu kotha to hol je if you are talking about cell division, then there are so many phases. To double the number of chromosomes, kuntu phase ot colchicine dibo lagibo aru titia ki hobo? So we need to remember is, if we apply colchicine, we have to apply it in the metaphase stage. So metaphase stage ot jodi tumi colchicine di dia bahiror pora, tetia hole ki hobo? The chromosomes will not separate. মেটাফেজৰ পিছত কি আছিল? এনাফেজ আছিল। এনাফেজত কি হৈছিল যে ক্ৰোমোজোমছ বিলাক সেপাৰেট কৰিছিল, ৰাইট? সো সেইখিনি সময় যদি তুমি কলচিচিন দি দিয়া তেতিয়াহলে কি হ'ব যে তোমাৰ এনাফেজ হলে সেই ক্ৰোমোজোমছ বিলাক সেপাৰেট নহয় পেলাই কি হ'ব? ডাবল ডাবল হৈয়ে থাকিব। That is what is called as doubling of chromosomes, right? So 2 points to be precise. The chemical that is essential for doubling of chromosomes is colchicine. At which stage of cell division you have to apply? You have to apply generally in the metaphase stage, right? So we'll come back to this. Polyploid, or we are producing certain type of polyploid. What is the polyploid? That has more than 2 sets of chromosomes. More than 2 sets of chromosomes ami keneke produce korilo by applying certain types of chemicals called as colcicid, right? Examples of polyploids could be Brassica. Second could be Triticale. Now these examples are important. Out of this the second example is most important. So yate ami ki koriso je ami duta different plant we have made a cross and then we have created a new plant, right? So ebilak ki hoise that these are all man made. So we have the first example that is called as Brassica. Brassica is a cross between two species, that is Raphanus and Brassica. Raphanus sativus is a scientific name of radish. Our Brassica, it's a name of a family, kintu Brassica bahut bilak species thakibo pare. Which species we are talking about? We are talking about the cabbage, right? So manhue ki korile, they have crossed between a radish and a cabbage together and they have produced a new offspring, and that offspring is called as Brassica. Similarly, we have something called as Triticale. Triticale is important from examination point of view. So Triticale questions kya hai? The first question, it could be example of a man-made polyploid, right? So it could be a Triticale. Second is Triticale is a cross between Triticale is a man-made polyploid. It is a cross between wheat and rye, right? Wheat plant and rye plant duidal cross kori plant notun plant adal produce korisile aru he notun plant dalok ami ki boli koisu Triticale. So wheat and rye, etu question ahe, it's a cross between wheat and rye. And similarly you have to know the scientific name also. This is Triticum species. Triticum species boli kyo likhisu, because Triticum is a genus. Triticum or bahut beleg species hobo pare, and the most common species that we talk about is Triticum aestivum, right? Triticum aestivum or logoto cross koribo pare ba Triticum beleg species or logoto cross koribo pare, aru tar logot ki koribo, rye cross koribo. And what is the scientific name of rye? It is sickle cereal, right? Sickle cereally. So we have Triticum and sickle cereally cross kori pela ami ki paisu Triticale. So Triti has come from Triticum and Kelly has come from sickle cereally, right? So etu examination point of view important. Etu bostu monot rakhiba. The two examples, one is Brassica and the other is Triticum, right? Next is polyploidor are dutaman point important. So while we are going for making polyploid with odd numbers are always sterile, right? Odd numbers mane trisex hobo pare, 5x hobo pare, 7x hobo pare. So what we are talking about is, we are talking about increasing the sets of chromosomes, no? Our chromosomes or logot, we use the word diploid, haploid, right? Polyploid, etc. So when we are going for polyploid, jeti ami experiment kori asu, tetia what we will see, if the offspring has a odd number of chromosomes, tetia ale he offspring bor khodai ki hobo, sterile hobo. Duta twice and cross kori satar F1 generation, no? F1 to ki hobo, khodai sterile hobo. F1 ot ami ki koribo lagibo, we have to make it fertile, and in order to make it fertile, what do? We increase or we double the chromosome of F1. So in short, odd numbers or jiman chromosome sets thake hebilak organisms khodai ki hoy, sterile hoy, aru even number thaka chromosome sets bilak hebilak khodai fertile hoy. For example, if you are talking about twice n, obviously twice n is fertile, or twice x is fertile, and if you are talking about trisex, 5x, etc., these are always sterile. So to be twice x kobo para, 4x kobo para, 8x kobo para. So enuka dhoroner jinbilak sets thakibo hebilak khodai ki hobo, fertile hobo, aru odd number jiman bor thakibo hebilak ki hobo, sterile hobo. So this is one point that you need to remember. Second is how do you make polyploids? Are formed in two steps. Duta stepot ami etu produce koro. First stepot ki koro, we two different species of plants are crossed by hybridized. right? So ইহত দুটাক যেতিয়া ক্ৰস কৰিম তেতিয়া হলে আমি কি পাম F1 generation. This F1 generation or the progeny is generally supposed to be sterile, right? Karon odd number ase. This is the step 1. Step 2 ki korim je we double the chromosome number of F1, right? So just a way to show how do you go for allo polyploids. What we do, we take two different species. For example, we have A and B, right? A and B mazor cross korim jonto offspring produce koribo tak ami ki kom first F1 generation, right? A F1 generation bur ki hobo that they will become sterile because we know that odd polyploids are always sterile. So ihot sterile hobo, ihotok fertile koribo lagile, that is the second step we are discussing. What is the second step? We have to double the chromosome number of this generation. And how do you double it? Obviously by the application of colchicine. So this is all about polyploid breeding, right? So here what we have concluded is, we have concluded the 4 methods through which you can improve a crop. What were the 4 methods? It was selection, hybridization, mutation breeding and polyploid breeding, right? Okay, so ultimately ami discuss ki kori asilo, we are discussing the steps of plant breeding, right? So the first step was collection of variability, we have completed. Second is evaluation and selection of parents. So entire these notes comes under that heading. So third step of plant breeding ki asile? What was the third step? কি আছিল পুরা evaluation selecting of parents was the second one, no? Third to ki asile? It was test selection and testing of for superior recombinants. Selection for superior recombinants. Right? So what we'll do here is selection. আমি যিখিনি ক্ৰস কৰিলো, যিখিনি progenies আমি ফৰ্ম কৰিলো, সেই progenies or মাজৰ পৰা এটা আমি সিলেক্ট কৰিব লাগিব, right? Selection is made among progeny of the hybrids. Means what? There we have collected the germplasm, we have selected the parents, we have those 2 parents, and after crossing them, what we had found is we have got or obtained many types of progenies, right? Bahut offspring palo. Hey offspring bilakor pora kuntu offspring sobotse superior. This is what we have to see. Ar sobotse superior kuntu keneke ami bisarim. আমি তো গৈ পেলাই চকুৰে চাই আহিলেই নহব, no? We cannot be doing everything morphologically. What we have to do is, we have to sow that progeny for generations. বহুত ta growing season or grow কৰিব লাগিব, and ultimately will get a better recombinant, right? So the selection is made among progeny of the hybrids, and second point is, it is a crucial step, no? It is an important step hobo, and so scientific evaluation is required. So cross kori pelai ami ki korilu je progeny to form korilu, progeny to form korar pisot amak ki lagibo scientific evaluation lagibo, and that plant should undergo self pollination. Self pollination hobo lagibo, self pollination hoar pisot ako tak ami grow korim, and ultimately ami ki pam superior recombinant to pam. So what we need to remember, selection is made among the progeny. Jiman pur progeny hol ar he progeny bur ami kor pora palu je ami hybridization technique apply korilo, ar tar pora jimlak progeny hoise tar pora ami best to select korilu. Best buli keneke gom pam je the plant has been allowed to undergo certain types of scientific evaluations, and plus tak ami ki korisu bahut kita growing season or grow korisu, and ultimately we have seen or we have obtained the superior recombinant. After selecting this, right, etia ami experiment korilu ba hybridization korilu, progeny palu, right, superior recombinant eta palu. Eta superior recombinant should be certified. Jeneke, if you remember in the last class, what we have said is jikono bostu eta correct hoise boli kobole. For example, if you are writing an answer, whether that answer is correct or wrong, there should be someone who needs to someone who needs to evaluate your answer, right? So there is a role of a teacher. There tumi শুদ্ধকে লিখিছা নে ভুলকে লিখিছা কোনোবাইটো ক'ব লাগিব. So we are talking about certification, right? So superior recombinant is something that we have got, কিন্তু সেই বস্তুটো যে superior কোনোবাইটো ক'ব লাগিব, no? So we are talking about the last step, the fourth step কি আছিলে? Testing, release, freeze হৈ গৈছে এতিয়া सुना भैया कुछ फ्रीज हो गया, फ्रीज हो गया बोले हेरी तो এটা ঠিক আছে okay. So we have talked about this, right? What we said is superior recombinant eta ami ultimately palu, no? Keneke palu je season self pollination korilu progenitor majot, A into B will give you F1 to jodi bhal hoy tete ami tar majot self pollination korilu, self pollination kori pelai jintu progeny palu ako next hetu ami ki korilu bahut kita seasons or grow korilu, and what we have obtained is from our point of view we can say that is superior. But as I said kunubai certify koribo laibo jetia loke hei progeny to ba hei plant dal best puli kunubai certify nokore hei bostu to tumi commercial purpose release koribo nwara. So in that case the fourth step is testing, release and commercialization. Right? So এই বস্তুটো কোনে কৰে? Right? You have a corporation called as National Seed Corporation. A National Seed Corporation or kam ki that they will certify. Right? What they will certify that suppose hey ami plant dal produce korle hey plant dalor juntu seed ase hey seed to prai to next baby plant dal grow koribo. So hey seed to say it's the best aru it has all the desired qualities kune check koribo or kune certify koribo? NSCA. Until and unless NSC certifies that seed ami hey seed to ketio commercialize by release kori dibo nwaru. So once they get certified, after certification, after that ki hobo? Release. Right? So egita asile the steps for plant breeding, right? So plant breeding ki hoy? It is a method through which crops has been or we are trying to improve the variety of crops, right? Aru improve ami kiyo koribo bisarisu because ekdom simple answer ki shortage of food problem to naikia koribo lagile we want more food, and so in order to get more food from the plants that we have, we want the desired qualities to be present in that plant, and for that we are going for something called as plant breeding. What are the steps of plant breeding? It's a long technique, and what are the steps is we have discussed it under 4 headings, that is collection of variability, selection of parents, then we have talked about testing or we have to choose the superior recombinants, and so হৈ যোৱাৰ পিছত কোনোবাই certify কৰি দিব লাগিব যে এই seed to it's perfect now, right? So these are the different steps of plant breeding. কিবা doubts আছিল নেকি? Okay. So within this chapter, what is the next thing that we are going to study? Out of all the methods that science has developed to solve the problem of shortage of food, one method that we have studied is plant breeding, but there are certain other methods also. So the second method is something called as green revolution. Actually methods বুলি ক'লে green revolution is not a method of crop improvement, but this is a phase, right? Etu enukwa eta phase juntu phase on production of some specific crops has increased, right? Drastically increase korise, drastically increase kora karone food shortage or juntu problem, he problem to some extent it has been solved, right? So since we are talking about plant breeding, the next point that we have to talk is about green revolution. So what is green revolution? Ki hoy green revolution? Green revolution or ketaman point. So before going into details, ekdom shortly but simply, whenever we hear about green revolution, what is the thing that comes to our mind? Ami ki buji pao green revolution, kisuman terms ase na, blue revolution, green revolution, white revolution, right? So green revolution mane ki increase in green plants or increase in plants is called as green revolution. And whenever we are talking about plants, we talk it from the sense of food, right? So যেতিয়া food increase whole from the plants, that is what is called as green revolution. Similarly, we have something called as blue revolution. Increase in what? Increase in fishes is called as blue revolution. White revolution ki increase in milk is called as white revolution. So we'll be talking about green revolution. Yare kisuman point. While we are going to write certain points, but from examination point of view, kuni eta point besi important. Yar pora ki question ahe je green revolution kuntu time ot arambho hoisile. You have to remember the year, not exactly the year, but mid 1960 is something that you need to remember. The second point is father of green revolution. The father or Indian father, duijonor kotha janibo lagibo. The third and the most important is some of the varieties of food produced because of green revolution. Whenever we are talking about green revolution, duta plant was of utmost importance, and what are the two plants we are talking about is always wheat and rice, right? So increased amount of wheat or increased amount of rice iman besike bahile within a specific period, and that is what is that phase is referred to as green revolution. So questions ahibo je wheat or ketaman variety naam koa, rice or ketaman variety naam koa, right? Aru green revolution kiho karone hoise? Karon wheat aru rice or content to besi hoi goise. Wheat or rice content kiho karone besi hoisile? Because we have a term called as semi dwarf variety, right? At a particular gene kisuman plants ot induce kora hoisile, and what is the gene we are talking about? We are talking about semi dwarf gene, right? So he dwarf variety adal plant jiman ukho hobo lagibo tatke ki korisile short kori disile, jeta short kori disile tar production besi hoi goisile, and that production led to this called as revolution, right? So green revolution, it refers to duta point janibo lagibo. Introduction of high yielding variety HYV. Remember this, high yielding variety of seeds. So green revolution at ki korisile? High yielding variety of seeds ki korisile? Introduce korisile. Next is increase in, it also led to the increase in the use of fertilizers and irrigation. So jikono eta revolution hobo lagile kiba to kam koribo lagibo no. So yate ki hoise green revolution ta mane food content to borhai dise. Food content to borhabole ki korisile je high value seeds bilak ami introduce korisile. Introduce kora mane what we understand, we have tried to plant or we have tried to sow only those seeds that have high value. High value in terms of what? High value in terms of yield, right? It might be in terms of growing period, je hunkale grow kore, hunkale fruit form kore, hebla ki hoy good qualities, no? So enukwa dhoroner plants ba enukwa dhoroner seed jar niki high yielding variety bilak ba high yielding quality asile, heblak ami grow korisilu. That is what is called as green revolution. Similarly, it also led to the increase in the use of fertilizer and irrigation. Fertilizers basically disile jate niki production basically hoy. So ei dutar basic meaning ki hoy, the green revolution was involved with introduction of HYV and introduction of fertilizers and irrigation. The second point is the phase of increase of two crops, especially wheat and rice, during mid 1960 is called Green Revolution. So Green Revolution ki hoy? It is nothing but a phase. Hey phase to ki hoisile? The food production increase hoisile, and food buli kole there might be so many various types of food, but to be precise we are concerned with the increase of the wheat and rice, because we know India at maximum ki khai? Eating wheat or rice, these are the most staple food, no? Eta beltor karone wheat is the most staple food, or eta beltor karone ki hoy? Rice is the most staple food. Out of all other foods, these are the two most important food. Egitai jetia bahisile, hey phase to kami ki buli koisilo? Green Revolution buli koisilo. So you need to remember wheat and rice. Since we are talking about increase in food plants, we have to talk something about agriculture also. We know India is an agricultural country, right? Right? India is an agricultural country. Yar bhitor ki monot rakhibo lagibo je agriculture contributes about 33% of the country's GDP. 33% of India's GDP. Etu point opera question ahe je kiman percent of the GDP agriculture cover kore. GDP full form ki? Gross domestic product. Yeah. So GDP kiman 33%. Second is agriculture, jehetu major thing in India, it provides employment also. It provides employment. Ako yara number to janibo lebo, to almost 62% of the population. Right. So since India is an agricultural country, what we need to remember is agriculture upor maximum India is based upon, right? So 33% of India's GDP, question 33% upor ahe, plus employment, almost employment to 62%. So as far as these figures are concerned, these figures as of now the figures has increased, right? So employment to be latest is what the percentage to obviously basi hobo, it might change. So these figures might change according to the latest information that is available, but we will be sticking to the information that is given in your NCERT book only, right? Because questions ho hey NCERT book ot jungita percentage dia ase okol hegita upore question ahibo, etia dhora yatke besi hobo pare komo hobo pare, but we would be sticking to 33% and 62% only, right? Next point. During 1960 to 2000, another figure that you need to remember, there was an increase in the production of wheat and rice, and rice. If we are talking about wheat, the production of wheat increased from 11 million tons to 75 million tons. Similarly, if you are talking about rice, it increased from 35 million tons to 89.5 million tons, right? So if you see the figure, there was a drastic increase in the production of two major food, that is rice and wheat. This increase is nothing but or the credit goes to green revolution. Ar jehetu besi increase ho goise tak ami ki boli khom green revolution kihor karone increase hol. All of a sudden wheat or rice or je 11 or pra go pla 75 million tons ho gol, rice or je 35 or pra 89.5 million tons ho gol, iman je besi production hol, there should be a reason behind, right? What is the reason? Reason to ki? Can anyone can tell me je iman bahile keneke? Kibata to obviously kibata hoise someone is responsible. So what is responsible is, now we have to talk about the contribution of that particular gene or that particular plant that is called a semi-dwarf variety, right? So increase kiho karone hoisile? This increase was due to the introduction of semi-dwarf varieties. So what did humans do? They have developed some new varieties of wheat and new varieties of rice. Aru he new varieties bilakok ki koisile? Semi-dwarf variety buli koisile. Dwarf is something that we understand, right? So jiman ukho hobo lagisile wheat plant dal he khinitke jodi okoman sapor ke bonai the plant or ki korise gene structure to change korise, gene structure change korar pisot what they have done is they have tried to produce a plant that is comparatively shorter than the original plant, and that is what is called a semi-dwarf. This type of semi-dwarf varieties has been made both in case of rice and also in case of wheat. And what they have noticed is, jetia semi-dwarf variety introduced kori dile, tahator production to obviously bahut besi asile as compared to their parents. So the entire contribution for green revolution goes to whom? Goes to the semi-dwarf varieties of rice and wheat. Rice and wheat are the two words that you need to remember, and together with that semi-dwarf is a word that you always need to remember when you are talking about green revolution. So here figures bilak monot rakhiba, semi-dwarf a word to monot rakhiba ne. Next is information point of view pra aru ketaman point. So now you have to talk about the father of green revolution. Right? Father of green revolution or Indian father of green revolution, you have two names of two scientists. So bhalke question to poriba, then you will answer. Father of green revolution, who is considered as a father of green revolution? Kakko? Dr. Norman Borlaug. Right, spelling sama, and you have to remember. He was an American agronomist. Right, so naam to mono rakhiba. Next is Indian father of green revolution. Ki asile MS Swaminathan. So he was the one to work upon wheat and rice during that period, right? 1960 jikhini samoyt India sobotse besi production of rice and wheat hoisile, and who was the man behind? The man was obviously Swaminathan, and so he is considered to be the father of green revolution or Indian father of green revolution. Next is, now what we know is green revolution kiho karone hoisile? The credit goes to something called as semi-dwarf variety. Semi-dwarf variety of what? Semi-dwarf variety of wheat and also of rice. So some information about dwarf variety you need to know. Dwarf variety or semi-dwarf variety of kihor asile? Wheat or asile or rice or asile? So edal wheat plant tomar normal height or pra jodi semi dwarf but dwarf koribo lage, what you need to do, you have to do something with the gene. So one particular gene was responsible for the production of that semi dwarf, right? So what you need to remember is, what was the name of the gene? Wheat or kuntu gene was responsible for production of that semi dwarf variety. Similarly, in case of rice also, which gene was responsible to produce that semi dwarf variety, right? So in case of wheat, we need to remember the gene that was responsible was called as Norin 10, and this was a gene that was reported in Japan. right? Next point. Semi-dwarf wheat. A particular wheat dalor kisuman specific characteristics asile. Ki characteristics asile was of high yield. Right? Anyway, so we are talking about crop improvement. Adult plant improve hoise ne nai. There are certain characteristics on the basis of which we analyze whether that crop is improved version or not. And what are the characteristics? The characteristics were, as we have discussed before, ki characters of base kori pelai ami sao je adult plant increase hoise nai ba better hoise nai. That is first point that we talk about is always about the yield, no, the production bahise nai. Second point might be about resistance. Resistance to what? Resistance to pest, resistance to environment, right, or unfavorable environment. And the third point that we talk about is the growing period, right? Jeti gute kita thake, for example, adult plant which has a high yield, it is resistant to disease and unfavorable environmental conditions, and plus it also has a shorter life, shorter life nohoy, obviously it has a shorter growing period, hunkale hi fruit produce koribo pare. So these qualities define a plant to be the best version of a plant. Similarly, in case of a semi-dwarf wheat, they also had those qualities. And what are the qualities? High yield asile, resistance asile, resistance to common pest, and plus it had shorter growing period. Right? So based on this, the plant was said to be a very good variety of wheat. Next is what we need to remember is certain examples of these varieties, no? Wheat or kisman semi-dwarf variety example. Examples of semi-dwarf variety of wheat. We have a term called as on a plant whose name is Sonara 64. Next is we have something called as Larma Roso 64. Eblak ki hoy wheat varieties, right? Semi-dwarf wheat varieties bur kisuman asile tar bitor example hol ei duta. Tar bitor aru kisuman ki janibo laibo. We had certain examples called as Kalyan Sona. The fourth example could be Sonalika. So these are examples of semi-dwarf variety of wheat. As I said, question 2 hodai enike ahe. They identify the semi-dwarf variety of wheat and identify the semi-dwarf variety of rice. So you have to remember the name Sonara 64, Larma Rojo, Kalyan Sona or Sonalika. Right. Another point of interest is when we are talking about Sonara 64 and Larma Rojo 64, a duta semi-dwarf variety actually code produced hoisile. It was produced or it was developed in Mexico, right? Dr. Norman Borlaug, what he has done is he has created the semi-dwarf variety, and name of that two semi-dwarf variety of wheat was Sonara 64 or Larma Roja 64. Etu code bonaisile Mexico, and from Mexico it was introduced to India. So tarpra loe nisile India jataniki it can grow, but what happened is they have seen that as far as Sonara 64 and Larma Rojo 64 is concerned, tahate Mexico environmental condition karone they could grow, but they cannot grow in India. So in order to make them grow in India, tahatok ki korisile, tahatok modify korisile. So these two were modified by gamma radiations. Right? Mutation kori disile, mutation kori kiho karone mutation koribo doka ba mutation breeding kiyo koribo laga hol, because they were not adjacent to the climate of India, right? And so they were modified by gamma radiation. You need to remember modified kihere modify korise hetu no remember koribo laibo, aru modify korar pisot tahate jundal plant pale he plant dudalor tahate notunke naam dile. Right? So wheat variety boli kole Sonara 64, Larma Rojo 64 Indiat kintu goza nasile because it was not suitable for the Indian environment, right? So they were modified and later they were named as Sharbati Sonara aru Pusa Larma. Right? So ekhini hol something about the semi-dwarf varieties of wheat that you need to remember. Tar bitor egita korpra anisile Mexico. From Mexico it was brought and then introduced in India. Introduced in India, right? So you have to modify kori, you have to remember these two names also, Sharbati, Sonara, Aru, Pusalarma. So ultimately we have got certain names, a name bilak ami from examination point of view you need to remember. So these are some informations about the dwarf variety of wheat. So because of these, the production of wheat increased drastically. Similarly, we have to talk about semi-dwarf variety of rice also. Dwarf variety of rice or semi-dwarf variety of rice. So jeneke this one jeneke wheatot ki hoisile je a gene was responsible. Similarly, in case of rice also there was a particular gene that was responsible, and the name of the gene was DGOU gene, right? Gene to naam monot rakhiba. Agortot ki asle when we are talking about wheat, the gene responsible was Norin 10, and when we are talking about rice, it is something called as DGOU gene. And Norin 10 was reported in Japan, and this was reported in Taiwan. Reported in Taiwan. The next point is this gene was introduced in rice plants by IARI. What is IARI? Indian Agricultural Research Institute, right? So tate ki korisile he modify korisile gene to ani pelai notun planter bhitor incorporate korisile, and as a result what we have obtained is we have obtained those dwarf varieties of rice. So example of semi dwarf variety of rice. Jaya and Ratna. Right? So these are the informations about wheat and rice. So what we have concluded is we were talking about green revolution. Green revolution at ki hosile drastic increase in food production, and which food to be precise. So what se basic kuntu food or increase hosile, that was in case of wheat and rice. And who is responsible for the increase? The semi-dwarf variety was responsible for the increase in production. Semi-dwarf variety of rice, for example, or semi-dwarf variety of wheat, for example, is something that we have discussed. Similarly, when we are talking about rice and wheat, we have to talk about another plant that is called as sugarcane, right? So sugarcane oto ki hoisile je tatu plant breeding kora hoisile, right? Now the point is sugarcane breeding kyo koribo laga hoisile. We have to talk about two species of sugarcane. Okol rice aru wheat is increase asile tenukwa noho. We have or they have done experiments on various other plants. Kisuman millets, kisuman cereals, kisuman rice, wheat, sugarcane. There are so many plants, but maximum production increase always in rice and wheat. We're talking about sugarcane. Sugarcane or duta species we have to talk about. Scientific name ki? Saccharum, right? So we will talk about two species, Saccharum barberi or Saccharum officinarum. Right, it was a sugarcane, right? A sugarcane tour, what are the points that you need to remember? This was something that was found in North India or grew in North India. right? This variety of sugarcane was poor in yield. It had a poor sugar content, right? Whereas if you are talking about this one, Chaccharum officinarum, this was a variety that was found in South India. It was obviously a better variety with high sugar content. A sugarcane dal bhalne beha productive hoy ne nohoy depends on what? Sugarcane, whether that sugarcane plant is productive or not depends on only one specific characteristic, that is if you are talking about the content of sugar that is obtained from that plant. Sugarcane plant dalor jodi sugar content e kom thake, then obviously that plant is not productive as compared to the plant jot niki sugar content besi thake. So if you are comparing between these two species, etar ki asile je poor in sugar content aru etar ki asile high sugar content, and plus another point of this species was it had thick stems. Obviously sugarcane dal jiman hokot hobo ba jiman thick hobo tar stem dal tar pora tomar sugar production besi hobo. So two varieties of sugarcane, one in North India and one in South India. Now what did the people of North India want? They wanted that such a good variety of sugarcane should be grown in North India also. Jihetu okol South India te iman bhal variety to grow korisile, so North Indians also wanted that this variety of sugarcane should grow in North India also. So what they have done is they have collected this species and they have tried to grow in North India, but to their surprise what they have seen is this species could not grow in North India. Obviously there were certain characteristics like the soil was a factor, the environment was a factor. So ebilak factors or karone South India junto iman bhal sugarcane variety to grow korisile but species to grow korisile, they could not grow in North India. So to solve that problem, what did the scientists do? The scientists, what they did is they made a cross between these two plants. Duida plant species by duida parent collect kori pelai tahator majot cross kori pelai they have created a sugarcane or they have created a progeny with a better quality, right? And ultimately that was introduced in North India. So sugarcane or ekhini information is anibolibo yar about questions maximum ki ahibo pare je kuntu species was found in South India, which species was found in North India. Similar, the next question that is followed is je kar sugar content bhal asile, kar sugar content kom asile. To aru solve koribo ki korile, hybridization was done between these two plants and a progeny was formed. So this is all about green revolution. So out of all the scientific methods, right, so what we have discussed is plant breeding, no, animal husbandry to discuss zoology class of koribo. Plant breeding we have completed, and one contribution or one most important point of this chapter is something about the green revolution and the plants, right, that were created. So we have done this. In the next class what we will be discussing is aru baki kita thaki gol, no, we have to talk something about single cell protein, right? I think we have an idea, no, single cell protein mane ki kisuman single celled organisms ase, those organisms could be used as food, right? So we'll be talking about single cell protein and something called as there are so many topics, tissue culture, energy crops, petroleum crops, etc. So up till here junkhini portion ami korisu dutaman questions we need to see. What are the questions or what are the type of questions that are asked from this section? So we will be discussing some questions. So asun kenuka questions ahe ekhini part opora. Okay. First question is hybridization between two varieties of same species is known as. Can you see the board properly? Intraspecific, intravarietal, interspecific, intrageneric. So this is the first question that has been asked from this portion. The answer is intra-specific, because we are talking about two varieties of the same species. The next question is, in mung bean, resistance to okay, this is something that we have not done, but resistance to yellow mosaic virus and powdery mildew were induced by. So resistance to disease, generally it is induced in a plant with the help of which method? The option is polyploidy, B is mutation, C is selection, and D is hybridization. Right? So resistance to disease is generally brought about by mutation. Third question is, Pioline variety of wheat. So you have to, this question has come from the example point of view. Wheat is NP836, PV18, Indore 2, Pusa Lerma, right? And the answer is obviously PV18. The next question is number 4. Tysome native 1, this is a variety of rice was developed in options: Taiwan, Japan, Mexico, Australia. So we are talking about rice, and this variety was introduced in Taiwan. So these are types of questions that has been asked from this part of the lesson that we have completed. Rest of the questions we can discuss in our assignment class, right? So just to have an idea that এইটো section of কি পঢ়িব লাগিব, these are the questions or this type of questions that you need to remember. And anyway, what is most important is obviously the examples that we have discussed. I think it's enough for today, right? So in the next class, we'll be starting with single cell protein. Thank you.